And we're recording, and we are presenting starting now. All right. So we've been talking about arthropoda. We've talked about the acari. That's the mites. We went over parasitic crustacea on Friday. What that leaves us with is insecta. Evidently, I need to fix that course number. That's a bit of a typo there, but we'll spend the last week looking at insects, lice today, fleas, and a couple of other really interesting and weird groups on Wednesday. And then that's it. We're actually coming to the end of a semester that I honestly have no desire to repeat. Not that you folks haven't been awesome, but, you know, dang. Right. So we talked about arthropods as being divided into chelicerates, which includes arachnids, crustaceans, myriapods, which are centipedes, millipedes, and a couple of others, which don't have any parasitic members at all, as far as I know, and hexapods, which includes the insects. As you might expect, hexapods have 3 pairs of walking legs. Hexapoda just means 6 legs in Greek. There's 1 pair of antennae. There's 3 pairs of mouth parts. And the tagmosis system is a body divided into a head, a thorax, and an abdomen. And appendages on the abdomen are highly reduced. So your typical insect will have 4 pairs on the head, the antennae and the mouth parts. The mouth parts may be very highly modified for different diets and different lifestyles, but what they fundamentally are legs, they're appendages. And then 3 pairs of walking legs on the thorax. And except in some of the earliest fossils that we have, there's no actual legs on the abdomen, except for the genitalia, which are usually kind of tucked inside, so they're not actually visible from the outside until the moment that the insect is using them for the established purpose. One of my favorite utterly useless facts is that to identify insect species, you often have to use genitalia shape. It's distinctive for different species. And one moment. Okay, morning, Sherpa. Okay, Sherpa, when you came in, we were just talking about insect genitalia, which I'm sure you really wanted to know everything about. Species of insect are often told apart by the shape of the genitalia because insects of different species often cannot physically mate with each other because the male's tallywhacker does not fit into the female's receptacle. Much like a key might not fit into a particular lock. And there was a company in Australia that made a very specialized device that was used by entomologists trying to identify insect species that would very gently inflate male genitalia so that they could be seen and drawn or photographed. Was called the phalloblaster, which I think is a really good name. And I think if I ever start up a punk band, I might call it the Phalloblasters or something like that. I think it's a really good name. Right. Genitalia are very highly modified appendages. They're usually kept tucked in. They're not usually visible externally. Sometimes the last segment of an insect's abdomen might have 1 pair of modified appendages called the cerci. And it seems appropriate that a town just a bit east of us in Arkansas is named after the things growing out of an insect butt. This kind of sums it up pretty well, the times that I've driven through there. True insects have mouth parts outside the mouth and have exactly 11 abdominal segments. And there are hexapods that are not insects that have mouth parts inside the mouth and don't have exactly 11 segments. The one that you're likeliest to have seen is the collembola, affectionately known as springtails. They tend to be very small, millimeter size or possibly less, but they're extremely abundant in leaf litter, in mosses, in soil. They're quite diverse, quite widely distributed, but not often seen unless you go looking for them. This just happens to be one from Britain called Neonura muscorum. They're called that because on the underside of the abdomen, there's a forked appendage that they can actually use as a lever to kind of push off and jump relatively very high very quickly, hence the name springtail. No parasites here. Insects, on the other hand, 1 million known species, something like 350,000 known species of beetle, 170,000 known species of moths and butterflies, although be it said, the vast majority are not large and not especially pretty. The number of beautiful butterfly species is maybe 1,000 or so. 125,000 known species of fly, almost as many known species of ants, wasps, and bees, and so on. There's a famous anecdote about the evolutionary biologist J.B.S. Haldane was giving a lecture on evolution, and at the conclusion, a sweet old lady stood up and said, Professor Haldane, what have your studies of the natural world taught you about the nature of God? And Haldane supposedly said, Madam, he has an inordinate fondness for beetles. God is clearly, if he made so many beetles, he's evidently a Beatles fan.
He's playing favorites.
Yeah, it's divine Beatlemania, as it were. Right. Okay. Thyraptera is the order of insects that includes the lice. The other big insect group that is entirely parasitic is Siphonaptera, which is the fleas, and we'll cover Siphonaptera on Wednesday. Lice are completely wingless. They used to be divided into Anapleura, which is the sucking lice, and Malophaga, the chewing lice. You'll still encounter those terms, but Malophaga is now known to be paraphyletic, meaning the last common ancestor of all of them was also an ancestor of all Anapleura. Anapleura primarily I think it only parasitizes mammals. We're the only taxon that gets sucking lice. Malophaga mostly parasitizes birds, but there are a few members that do parasitize mammals. The cat louse, Felicola, is a chewing louse, and so is one of the species of lice that domestic dogs can get. The other two are sucking lice. So sucking lice suck blood. Chewing lice feed on, if they're on birds, they'll feed on feathers. They feed on dead skin. On mammals, it's the same. They'll feed on, hair, dead skin. Sometimes they'll also eat living tissues, and sometimes they will also drink blood, even though they've got chewing mouth parts rather than the piercing mouth parts of anapleuran lice. There's the way you can tell them apart. On the right, that is a human, now I can't remember if that's a head or a body louse. If you go back way to the beginning, I actually pointed out that head lice and body lice in humans are possibly the same species, but in the process of beginning to separate. There are some differences between the two. Either way, that's a human sucking louse over there on the right. You can see it has a narrow head. Chewing lice, on the other hand, like the one on the left, the head is wider than the front of the first segment of the thorax, which gives them this kind of necked appearance. That wide head you can clearly see over there on the left. You can also tell them apart, of course, by the mouth parts. Chewing lice have jaws. Sucking lice have sucking mouth parts. Sucking lice really suck. Yes, I said that. There's only about 500 species of sucking lice, and I was surprised to find this out. Only about 20% of mammal species have sucking lice at all, and there's entire orders of mammals that don't have them. The monotremata is the platypus and the echidnas, and it's not a very big group, but it is perhaps interesting to know that there are no lice on these at all. Platypus don't get lice. Good for them. Sirenia is the manatees. They don't have lice. Neither do whales. You might expect that because manatees and whales are both marine, spend a lot of time in cold ocean, so it's probably not a very good habitat for lice. Proboscidea don't have sucking lice at all, although they've got another type of lice. There are at least 2 sucking lice that infest dogs. There's 5 that are known to infest cattle, and this happens to be one of them, the little blue cattle louse, Selenopotes capillatus. So you vet people might encounter these, possibly more likely in large animal practice, although you can get sucking lice on dogs. Oddly enough, one mammal group that does have sucking lice is seals. And I just said that whales and manatees don't get sucking lice because they, swim in the ocean. But seals actually can get them. In some cases, they live, since they're living on the skin among the hairs, fur is insulating because it traps air. It's not so much the hairs themselves that block heat transfer. It's because the hairs collectively trap this little bubble of air right next to the skin. And in some cases, these lice are actually living in this little warm, airy bubble, sheathing the skin of the seal or the sea lion, even though they're surrounded by very cold water on the outside. There are others that seemingly make a living that are much more exposed to the cold, salt water, and I'm not quite sure how they do that. But I did come across this Echinoptyrus horridus, a louse parasite of seals, and one of surprisingly few marine insects. There's these. There's a couple of marine water striders, that'll skate over the surface of the water, and those are insects. They're in the bugs, the true bug family, the true bug order. But there's a few flies that'll live in brine pools and things like that. But there's not many insects in the ocean, and this is one of the few. I would guess that these guys are related to the lice that parasitized, the common ancestor of carnivora, dogs and cats and bears and weasels and things like that. That would actually be really interesting to look at, if the lice on seals are more closely related to the lice on land carnivores than anything else. You'd predict they would, but I don't know. That gives me an idea. Okay. Those are the 4 groups that we divide lice into today. And third from the left, Anapleura, is the sucking lice. On the far right, Rhynchopthorina, that is a very oddball group of only 3 species that parasitizes elephants and warthogs. And I don't know an awful lot about them. I've never been close enough to a warthog to take a close look at these. It's not a coincidence that they've got this very long proboscis because they have to drill down through elephant hide. And as such, they've got this very long, drill-shaped, tube-like mouth parts for drilling through elephant skin and getting down to the blood. And then much more diverse are Amblycera and Ischnocera. These are the two main groups of chewing lice. There's a few members of both taxa that feed on mammals. Ischnocera includes one species on dogs and a species on cats, probably a few others on mammals that I didn't look up in advance. Amblycera can parasitize some South American mammals, but most Ischnocerans and Amblycerans feed on birds. Ischnocera are noted for being extremely host-specific. In some cases, they are restricted not only to a single species of bird, but a specific region of a bird. You might have 2 or 3 species on one bird individual, each one adapted for a specific body part. Amblycerans tend to be a bit less restricted. Amblycerans will feed on feathers, skin, sometimes blood, sometimes live skin, whereas Ischnocerans are feather specialists. And those chewing lice will spend their entire lives on a single host. And again, we say that's their monoxenous. They can be transmitted from parent to offspring. You know, when mommy bird is sitting on her babies in the nest, some of the lice will hop off. Sometimes lice can hitch a ride on flies. This is a blood-sucking fly known as a ked. And I don't think small animals get these, but livestock get keds. Sheep keds are particularly nasty parasites of sheep. And lice have been known to hitch a ride on hippoboscid flies. And remember, we call this foracy, this hitch a ride relationship that you sometimes see. So there you've got a bird louse hitching a ride on a hippoboscid right there. You can see the louse down on the left side of the abdomen. And as I said, chewing lice can be extremely host-specific. California condors were highly endangered, and I think we're down to, something like 20-some individuals at one point. Now, when it was decided to actively try to save the species, they were all captured, taken out of the wild, and kept for a captive breeding program. And when the last wild California condors were captured, they were deloused, and that wiped out Colpocephalum californicae. That's a species of chewing louse that lived absolutely nowhere else. So California condors have been returned to the wild. The population has grown and stabilized, and they are completely louse-free because when they went through that bottleneck, we wiped out Colpocephalum californicae. So that's another species that died out because of direct human intervention. One of the few species I think humanity has deliberately tried to kill. And because these chewing lice are so host-specific, when you get bird populations dividing, louse populations divide with them. And the upshot is that the cladogram of bird lice often matches almost perfectly the cladogram of birds, because whenever a bird species divides into 2 new species, the lice on the birds will divide into 2 new species of lice. So here's a neat example of this. This is Digariella regalis. It's a chewing louse, and its natural host is Buteo galapagensis. Buteo galapagensis is the Galapagos hawk. It's in the same genus as red-tailed and red-shouldered hawks, which you may know. I've seen both of them around Conway. They're not that difficult to spot around here. And both Buteo galapagensis and its closest mainland relative are parasitized by Digiriella regalis. Now, the Galapagos is this group of islands 600 miles off the coast of Ecuador, and they're famous in evolutionary biology because Darwin stopped for a visit in 1835, and he noted that they had very distinctive life, but the species on the Galapagos were oddly similar to mainland species, yet different in some critical ways. And this was one of the things that he drew on in developing, the theory of biological evolution. The idea was that Galapagos species had colonized the Galapagos from the mainland. Maybe they'd gotten blown off course in a storm or something like that. Doesn't really matter how. And as they'd colonized the Galapagos, they'd adapted to the new habitat in the Galapagos, the new food supply and new environment and so on, and had ultimately adapted so far that they had become separate species. I assume you guys have heard this story before. Now, the Galapagos hawk is descended from American hawks. Swanson's hawk is the closest relative. It's descended from American hawks that colonized the Galapagos. It lives on 8 of the islands, the ones that are color-coded here. And populations of the Galapagos hawk rarely mingle because hawks don't like to fly over open water. So we not only have a separate species in the Galapagos of Galapagos hawk, we've got 8 populations of Galapagos hawk that probably still can interbreed, but rarely do. And we might predict that if left alone and no disasters happened, that eventually we would have 8 separate hawk species in the Galapagos Islands. That hasn't happened yet, but we still do have 8 genetically diverse populations of Galapagos hawk that are distinctive from each other. And in a study in 2014, they took hawks and they took their lice and sampled mitochondrial DNA. Now, what you can do with mitochondrial DNA is assign your sequence diversity to various clusters, and then you can assign individual hawks to DNA clusters based on what DNA alleles that they've actually got. And that's what these diagrams show. Most of the Fernandina hawks have most of their DNA belonging to a cluster that is depicted in blue, although there were a couple of Fernandina hawks that have sizable ancestry coming from a cluster that is shaded in orange. And then for Isabella, it's the same. If I'm going from left to right on these diagrams, and that first grouping labeled Fernandina, that's mostly blue, what that is each one of those vertical bars represents a hawk, and the colored zones is basically the proportion of allelic diversity in each Fernandina hawk that can be assigned to a particular cluster. So most of the Fernandina hawks, with a couple of exceptions, have DNA diversity that belongs to a blue cluster, a particular cluster that's shaded blue. Most of the Isabella hawks have DNA diversity that belongs to a cluster that's shaded orange, although there are some hawks that are assigned blue in Isabella. And if you look, Fernandina and Isabella are right next to each other, so that is perhaps not surprising that there might be some interchange between the Fernandina and the Isabella hawks. Santiago, you've got a couple of clusters that are present there. And then on Marchena, Pinta, Santa Fe, and Española, those are smaller and more isolated islands. And almost all of the hawks on those 4 islands have DNA sequences that are assigned to unique clusters. So if we go back, you can see Pinta, Marchena, Santa Fe, and Española. They are far from the large islands of Fernandina and Isabela. Hawks flying from Fernandina to Isabela is doable. From Isabela to Pinta, much more difficult. And so almost all of the hawks on those 4 islands have sets of alleles that can be assigned to these distinctive clusters that are found almost nowhere else. So you're just looking at a diagram, very colorful, color-coded diagram, showing you how much of a hawk's ancestry can be assigned to a particular DNA cluster. And you can see that Marchena, Pinta, Santa Fe, Española, very well separated. And the really cool thing is if you look at the diagrams below it, almost exactly the same thing is true for lice DNA. Not only are the Pinta Galapagos hawks very distinctive in the particular set of alleles that they've got, the lice of the Pinta hawks are also very distinctive for the alleles that they've got. And the same for Marchena and Santa Fe and Española. So we've got several hawk populations that are genetically distinct from each other and that rarely interbreed and that maybe given time would be becoming separate species. And their lice, it's exactly the same thing. We have several louse populations that are distinct from each other that very rarely interbreed and are on their way to becoming different species. And incidentally, they've looked at fitness of Galapagos hawks. Galapagos hawks are territorial. You'll get a family group of birds that will stake out a territory and defend it. We actually talk in evolution class about this. The Galapagos Islands are actually pretty harsh. They get severe droughts every time there's an El Nino year. Even when that's not happening, they are volcanic. The water surrounding them is fairly cold. These are not lush tropical islands where you can sit back and drink fruity rum drinks with paper umbrellas in them. These are quite literally desert islands. Darwin actually came surprisingly close to death once, taking a hike across a big lava plain on one of the islands. It's not an easy place to make a living, and the best way to do it is cooperatively. So you'll get these family groups that will stake out a hunting territory, feed there, and defend it against all comers. There are some hawks that are non-territorial, but they tend to be in worse physical shape. I'd have to look up exactly how, but they tried to measure physical condition shown by the y-axis. And you can see non-territorial hawks tend to be on, below the 0 line. On average, they're in worse physical shape than the territorial hawks. And the non-territorial hawks also have more lice, between 4 and 7 lice on the body, as opposed to most of the territorial hawks have between 0 and 4. Off the bat, I'm not sure what's cause and what's effect. I'm not sure if greater louse infestation decreases body condition or if poor body condition allows more lice to infect you. But you can see at the very least there's a correlation between lifestyle, territorial or not, physical condition, and abundance of parasites. Even if I don't remember off the bat which way the causality on this is supposed to work. Anyway, take the story about lice on Galapagos hawks and multiply it. On the Galapagos hawks, we have these distinct populations of lice that are specific to distinct population of birds that might, given enough time, speciate. Blow that up, and you can see the same thing happening in sizable groups of species. On the left, you've got a cladogram of pelicaniforms. That is pelicans and anhingas and boobies and frigate birds. That's the phylogeny over there on the left. And then on the right, there's a phylogeny of their chewing lice in the genus Pectinopigus. And they're almost exact mirror images of each other. There's only one point. If you see that place where lines cross, there's kind of an X in between. There's only one point where it looks like there might be a host switch. Everywhere else, every time a common ancestor gave rise to two descendant species of bird, the common ancestors lice gave rise to two descendant species of lice. The phylogenies map onto each other nearly perfectly. So that's something I've always appreciated about chewing lice. That's not a sentence I ever thought I would say. Okay, questions, comments, rotten fruit, except you can't throw it because like you're not here.
What about our final? Like, are we still on the 6 prompts pick some track?
Yeah, and I've got I just got an idea about a really good question to ask.
Ew.
Yeah, afraid so. Yeah, I will. I didn't get anything done this weekend except for some honeydews, but yeah, I'll actually have some time this afternoon to sit down and actually come up with something to give you goobers.
Will you be kind to us? As kind as you can be?
I'm just thinking at the beginning of the semester before all of this stuff went down, I kind of enjoyed playing the role of the mustache twirling sadist. But I've never been very good at actually being sadistic, and now it just, would seem even more inappropriate. So, yeah, I will try to come up with interesting questions that are nonetheless well within your ability to solve.
Thank you.
And I suddenly remember that I haven't uploaded Friday's lecture, so I guess I will do that and this one. I'll get those both up today. How long will you have to do the final? I'm envisioning it as being, again, not full bore term paper, but, slightly longer essay questions that you will have to go, look up some outside sources for. And I'm going to try to give you guys at least a week.
Another question.
Say again?
I have another question. Are we going to get our grades before finals start for like our current grade in the course?
I'll give you everything I've got.
Sweet.
All right. Oh, I've been recording all this time. I'm going to stop now.